And today, as I said, I'll explain mitosis, and then after I'll explain meiosis. In mitosis, the other time we said that basically a parental cell divides to form two daughter cells. Daughter cells are two which are formed, and these two daughter cells are having exactly the same number of chromosomes as the parental cells. So if you're defining mitosis, make sure you talk about the two daughter cells which are formed and they're having the same number of chromosomes as the parents. This process always takes place into basically 4 stages, but we consider 5 because it occurs after the resting stage called the interphase stage. So the moment interphase takes place, the first stage begins for this process, that's called prophase. Another stage that follows is called metaphase, anaphase, and then lastly telophase, which is followed by cytokinesis. To remember the order of the stages, we just use the word EMMAR. That is interphase, prophase, metaphase, anaphase, and telophase. Now, in each of those stages, we have events that take place. For example, in this stage that we say to be the resting stage, we have some other events which take place. So when we are talking about interphase, it is sometimes described as the resting stage. During this stage, there is often making of proteins, because proteins will be needed when the cell is dividing. So one of the events, you say, protein synthesis occur. Then some organelles, remember when the cell is dividing, some organelles will go in the other cell and the others will go in the other cell. So another event is formation of organelles. After organelles have been formed, there is formation of DNA. So replication of DNA, such that the DNA becomes to be 2. It also builds up sufficient energy store. So you say there is formation of energy that will be used in the process. Remember, the process of protein synthesis occurs with a lot of energy. So after interphase has occurred, we enter into this process of mitosis. So to go through this, during interphase, the chromosomes are not visible. You cannot see these chromosomes. Chromosomes can only become visible under microscopes during the process of cell division. So under light microscope, just before mitosis begins, the centrioles are among the most prominent organelles. So it is very important to take note of the events which take place in interphase before we enter into the real meiosis process. So the stage that follows that is called prophase. Now prophase is divided into what we call the early prophase and the late prophase. Early prophase, that's the beginning of prophase. At first we say that in interphase these chromosomes are not visible. So we see that the chromosomes become visible as they contract. So when they contract, the nucleus shrinks. You will see that the centrioles will be moving to opposite poles, as we shall see in the diagram. The spindle fibers will start to form. These are the spindle fibers, these ones, and this is the this is the centriole, and this is the centriole. These are the chromosomes, and this is the nucleus. So the event you should note here is that the chromosomes contract, the nucleus shrinks and disappears. The centrioles move to opposite poles of the cell, and then in late prophase, chromosomes become shorter. I think you can see them, they are now very short and fatter. You will see that each chromosome is composed of two chromatids that are held at a position called centromere. So there is always what you call the nucleus in a nucleus, where the ribosomes are made. This nucleus disappears, and prophase ends with the breakdown of the nuclear envelope. So after that is done, we enter into another stage. The stage is metaphase. In metaphase, the chromosomes, they just come and arrange themselves at the equator. If this is a spherical cell, then this is its equator. So the chromosomes arrange themselves at the equator of the spindles. So these are the spindle fibers. So the chromosomes have come and they've arranged themselves, and when they arrange themselves at the equator, we enter into another stage called the anaphase. In anaphase, the chromosomes separate and form what we call chromatids, and these chromatids are moved to opposite poles of the cell. We can see that these chromatids being pulled to this side, and this chromatid is being pulled to this side. So that stage where we have the separation of the chromatids in mitosis is called anaphase. And then after, we enter into the last stage called telophase. In telophase, the chromosomes, those chromatids reach at the equator, meaning at the opposite poles, and when they reach, they become chromosomes. Now the things which were disappearing in the prophase, they now again reappear. For example, the nucleolus reappears, the nuclear envelope again reforms, again the chromosomes which shortened and became visible, now they become very thin and you cannot see them. In fact, they form what you call the chromatin, the coiled form of chromosome. That marks the end of telophase, and then we enter into another stage that we call cytokinesis, where the cytoplasm divides, and then later the cell membrane, and if it is in plant cells, there is just formation of what you call a cell plate. Now to summarize, these are the stages we've been explaining. That in interphase, you will never clearly see these chromosomes. You will only see their nucleolus, the nuclear envelope is there, the plasma membrane is there, and now the centrioles are also within the cell. Now after that interphase, we enter into what we call in prophase. In prophase, we can see that these chromosomes, they have now shortened and they are visible. You can see that each is having two chromatids held at the centromere. From there, we have the centrioles. These ones, they have now moved to opposite poles, and we see that the spindle fibers have been formed during the stage. This is the early prophase and this is the late prophase, the one we are calling the metaphase. Then after that has happened, they come and arrange themselves at the equator of the spindle fibers, and the stage is called the metaphase. After they have arranged themselves, they split, they break into chromatids in anaphase, and they are separated, they are pulled apart to opposite poles of the cell. After that, we enter into what we call a telophase. In telophase, we say that they reach, and when they reach there, they become chromosomes. Spindle fibers have disappeared. The nucleus comes back, the nuclear membrane and the nuclear envelope also comes back, and cytokinesis begins. Cyto is cytoplasm and kinase is division. So the division of cytoplasm. So when we look at the importance of cell division, we see that mitosis has played a role in growth. We cannot grow without mitosis. When you get a wound, repairing of it is done by mitosis. And even maintaining the traits within an organism, since there is maintaining the number of chromosomes and the daughter cells are always identical, there is that advantage that you can maintain a character that you desire. In meiosis, we shall also explain it after we have understood how meiosis occurs. Thank you. Next time we shall be watching meiosis. Please, you can subscribe to the YouTube channel such that you can receive whatever update is put on.